Brown Fat's Potential for Solving the Obesity Problem by Connor Crowley, Colin Him, Bruno Dos Santos, Elizabeth Conkey, and Jessica Bose. Introduction to brown fat. Brown fat. What is brown fat? Brown fat, also known as brown adipose tissue, is one of two types of fat in humans and mammals, the other being white adipose tissue. Brown fat's main function is to generate body heat in newborns and animals, mainly mammals, especially those that are hibernating. Mammals and humans with higher levels of brown fat take longer to start shivering. That's why most newborns don't shiver, because they have high levels of brown fat, while adults have low levels of brown fat. As humans get older, brown fat disappears and white fat increases. However, their functions are very similar. The key differences between brown fat and white fat is that white fat contains a single lipid droplet, while brown fat contains numerous small lipid droplets. In addition to the small lipid droplets, brown fat has numerous mitochondria that contains high levels of iron. The dark color of brown fat is also a result of the high levels of iron in the mitochondria. Brown fat generally has more capillaries than white fat, given that it has a greater need for oxygen than white fat does. These two photographs are brown fat, which is on the left, and white fat, which is on the right. You can see the main differences between the two, which is color and the number of capillaries.
The study Cold but Not Sympathomimetics Activates Human Brown Adipose Tissue in Vivo was conducted to determine what activates brown adipose tissue. In order to determine what activated brown adipose tissue in humans, 10 human volunteers were subjected to either a dose of ephedrine, mild cold, or a saline solution, which was the control. 18-fluorodeoxyglucose was used to measure BAT activity. The observations from the experiment were that the patients dosed with ephedrine had an increase in blood pressure, an increase in heart rate, however no measurable effect was made on the activity of the brown adipose tissue. For the patients exposed to mild cold, there was also an increase in blood pressure. However, there was a decrease in heart rate and substantially increased activity of the brown adipose tissue. Figure 1 displays the effects that the controlled saline solution, the ephedrine, and the cold had on the patient's blood pressure and heart rate. Figure 2 shows the amount of detectable brown adipose tissue that was observed after the experiment was conducted on both males and females. And Figure 3 shows the detectable amount of brown adipose tissue activity observed in the patients. As you can see, the patients exposed to the cold had a higher volume of detectable BAT as well as increased BAT activity compared to the other groups of patients. The results of the experiment were as follows. Exposure to cold substantially increases BAT activity as measured by the absorbance of fluorodeoxyglucose, while the weight loss drug ephedrine had no measurable effect on the activity level of brown adipose tissue in humans.
This is the title of the next case study. This case study was designed to determine if brown adipose tissue is metabolically active and whether or not it contributes to cold-induced non-shivering thermogenesis in humans. 6 test subjects were placed in suits and lined with tubing that was able to have water go through it to create a water bath to lower the body temperature. The results of the temperature case study showed that there was an inverse relationship between BAT activity and energy expenditure. And it also showed that a cold exposure stimulus enhances BAT oxidative metabolism. This is just a figure showing that the cold temperature of the body helped increase the radiodensity of the BAT in the body.
Retinaldehyde 1A1 is a key determinant of white adipose tissue plasticity and the regulation of white versus brown adipocyte characteristics. Retinaldehyde 1A1 is predominantly expressed in visceral fat, as shown in the graphs and charts below. Obese humans have more retinaldehyde 1A1 in their visceral fat tissue than lean humans. This evidence suggests that there is a correlation between retinaldehyde 1A1 and obesity in humans. Because of the relation of retinaldehyde 1A1 to obesity, deficiency in the retinaldehyde 1A1 enzyme causes greater energy expenditure in white adipose tissue. This energy expenditure by the white adipose tissue is similar in behavior to the energy expenditure in the brown adipose tissue. The energy expenditure comes from burning fats. This makes the deficiency of retinaldehyde 1A1 important for weight loss research. Retinaldehyde 1A1 deficiency activates a thermogenic program in white adipose tissue. Increased mitochondrial uncoupling drives oxidative phosphorylation, enhancing cellular respiration. Oxygen consumption rates increased in white adipose tissue and in visceral tissue, but not in brown adipose tissue in humans. Because of this thermogenic nature, rather than storing energy, white adipose tissue will release it, similar to brown adipose tissue.
The scientists then wanted to move towards gaining a better understanding of the mechanisms involved for this increased uncoupling in the white adipose tissue. So what they first did was took a cell line that was an acceptable model for adipogenesis, and they stimulated it with retinaldehyde alone and found that this induced uncoupling protein 1 gene and protein expression significantly. It was then shown that uncoupling protein 1 expression was increased the same amount when retinaldehyde inhibitor was present, proving that retinaldehyde induction of uncoupling protein 1 expression in white adipocytes was independent of its conversion to retinoic acid. Retinoids exert their action within the cell by activating nuclear hormone receptors such as RAR and RXR. So in a subsequent experiment, RAR antagonists were added in vitro, and it was shown to completely stop expression of uncoupling protein 1 and other known brown adipose tissue marker genes. Next, rats were injected with the RAR antagonist to further test whether this thermogenetic phenotype observed in the retinaldehyde 1A1 deficiency was mediated through RAR. During exposure to the cold, it was found that the rats that were injected with the RAR antagonist had lower body temperatures compared to controls, suggesting that the thermogenetic program in retinaldehyde 1A1 deficiency is indeed RAR dependent. In a subsequent experiment, it was also found that retinaldehyde recruited PGC1 alpha, already located in the mitochondria, and this transcriptional coactivator subsequently induced uncoupling protein 1 transcription. To further test the findings of this experiment, the mice were injected with a validated retinaldehyde 1A1 antisense oligonucleotide, which is basically a synthesized DNA strand that inactivates retinaldehyde 1A1. And this retinaldehyde 1A1 oligonucleotide showed decreased retinaldehyde 1A1 in RNA and protein levels in the liver and the white adipose tissue, but the levels were not changed in the brown adipose tissue compared to the control, and this significantly increased uncoupling 1 protein expression in white adipose tissue. The functionality of this was tested by placing mice in cold environments. The mice injected with retinaldehyde 1A1, the antisense oligonucleotide, were better at maintaining a constant core body temperature in the cold, and it was also found that the treatment with retinaldehyde 1A1 antisense oligonucleotide significantly limited weight gain in obese mice and decreased white adipose tissue mass. All this showed that retinaldehyde 1A1 repression in visceral fat induces a thermogenetic program that helps the mice to not gain weight and improve glucose homeostasis in obesity. So the take-home message from all these experiments is that 1. Exposure to mild cold causes increased brown adipose tissue activity, leading to weight loss, and in order to make white adipose tissue act as brown adipose tissue within the body, one method is the blocking of retinaldehyde dehydrogenase, and through various mechanisms this activates uncoupling protein 1, which promotes mechanisms in white adipose tissue similar to mechanisms found in brown adipose tissue, but without altering brown adipose tissue activity or function.
And here's our works cited. Thank you for listening to our presentation.